urea. No, sorry. It's liver, right? It's liver, it's not kidney. Sorry. It's liver. Remember, yeah? The organs produce the urea is liver, it's not kidney. Okay. So after the liver produce the urea, what will happen is the urea, right, they will be excreted out by the kidney. So means that the place which the place which the organs produce the urea is in the liver. So I'm not sure you still remember about this, right? Last time we learned about the digestions. Okay, where's my pen? Okay, let me show you the pathway for the protein digestions. So last time we learned about this one, which is the proteins. When the protein being break down, right? Actually, I do send this exercise to you already. You can check from this WeChat. You see, chapter 30, paper 2, past year. Yeah, you can download it. So protein, right? After they undergo the digestions by the enzyme, which is a protease, right? Protease is a kind of enzyme. Okay, you have this protease. So it will become what? Amino acid, right? Amino acid. So the amino acid will be produced at where? It's being produced at the intestines. Okay, and then it will be absorbed by the intestine villi. Do you remember? It will be absorbed by the intestine villi together with the glucose. Right. So the intestine villi, right, which is this one? The intestine villi, there is a blood capillary. Okay, which is found within this intestine villi. Okay, you can imagine that this is a blood capillary. So when the amino acid, right, they'll be absorbed by the intestine villi. Actually, they will enter into the blood capillary. They will enter to the network of blood capillary, which is surround the intestine villi. So now the amino acid, they get into the bloodstream. So they get into the bloodstream, right? So which is in the bloodstream. Bloodstream. Then what will happen is from the intestines, which part of organ they will go? From the intestine, they will reach where? And as well, which part of the organs they will go? After they get into intestine, right? And then for the intestine villi, they will enter into the blood capillary. So now they get into the bloodstream. So from the bloodstream, they will transport to which organ? Do you remember? It's the liver. They will get into the liver. This one, liver plays a very important role in the metabolisms of the, right? For example, like proteins, right? Yeah. For example, in the carbohydrate metabolism also happen in the liver. So that's why carbohydrate metabolism, they will help in do what? They will help to break down, right? It's not breakdown. It's converts. They will help to convert the excess glucose become glycogen. So the glycogen will be stored inside the liver. That's why in the liver you found glycogens. So liver, one of the function of liver is they will store the glycogens. Do you know why they need to store glycogen? If there's an excess production of glucose, and not all glucose will be used up in the cellular aerobic respiration. So there will be a remnant of the glucose, right? So this leftover glucose, they will be transported to the liver. So they will store in form of glycogens. Because glycogen is a kind of polysaccharide. Okay, I'm not sure if you remember or not. It's a polysaccharide. So polysaccharide is a very suitable energy storage compound because polysaccharide is formed by very large and compact structure, right? Yeah, these large and compact structures, they will not affect the osmotic pressure of the blood. So just remember that glycogen, right, is being stored inside the liver and also the muscle cell. So this inside, they have the glycogens, okay? There's a glycogen inside. Apart from that, they also have the amino acids. So this is, we talk about the protein metabolism. They also have the amino acid, right? Because just like we talk about during protein digestions, the protein is being break down by the proteins and then they will become amino acids and then they will be transported to the liver, right? They will get into here, okay? They will get into here. So amino acids, they will undergo what? They will undergo a process we know as the deaminations. So I'm not sure, do you still remember about the deaminations? Deamination is a process which is they will involve in the removals of the amino group from the amino acids. So it's something like this. This is the amino acid, right? Okay, amino acids. I hope you still remember their structure, right? So amino acid is like this COOH carboxylic group, and there's a one amino group NH2. And then there's a upper, this side will be the R group, which is the alkyl group. And then below side, oh, what happened to my pen? And then below side will be what? Will be the hydrogen atom. So deamination, right? Since they remove this one amino group, so they are actually remove this amino group here. So from the amino group, after they've been removed, right, they'll become what? They'll become ammonia. So ammonia is being produced as a product of the protein metabolism. This is known as ammonia. Ammonia, they have about NH3, they have 3 carbon atoms. So ammonia is being produced inside the body line. And then ammonia is toxic, is considered as toxic to the body. So your body, well, they will try to convert this toxic ammonia become something less toxic substance, which is the urea. From very toxic ammonia become less toxic urea. So this process, right, which is involved in the formation of urea, they are all happen in the liver. It's not in the kidney, but it's in the liver. Liver is the organs which they will produce the urea from the deaminations during protein metabolism. So after that, after the liver produce the urea, what is the next step? The urea will be excreted out by the kidney. Okay, so now will be the kidney. It will excrete out the urea. Okay, excrete out. So just remember that this one, I think I do put here, right? Okay, I just put here. This is very important concept. Liver produce urea. So the urea is being produced from the protein metabolism. So after the liver produces the urea, urea will be excreted out in the urine by kidney. So kidney is just an organs. They will help to remove the urea away, since kidney is a kind of is part of the excretory system, right? They will help to excrete any kind of the waste products, include the urea and also something like mineral salt. So this is the answer. Example, excretions, removals of the urea by the kidney. Okay, this one number C. Removal of the undigested food through the anus. This is referred to the ejections. It's not excretion. Ejection. Ejections is a process only involved in the removals of the feces. Only feces, right? Excretions is involved in the removals of the waste substance, includes those carbon dioxide, urea, mineral salt, and feces, and it includes feces as well. So the excretion, right, is considered as a characteristic of the living organism, but not include ejections, because not all living organisms, they do produce feces. And then for this one, in kidney tubule, which substance are filtered out of the blood in the glomerulus? So this is number C. So what does this mean? This means that, just imagine, right? Let me draw a glomerulus for you. This is something like glomerulus, okay? There's a lot of highly networked blood capillary. And then here will be the Bowman capsule. Then here will be the proximal convoluted tubule, and then here we will collecting. It's known as loop of Henle. So the glomerulus, right, actually is referred to the network of capillary at here. Just imagine that glomerulus is actually the blood capillary, which is surrounded by the Bowman capsule. So this is a glomerulus. It's not a structure, it's just a network of blood capillary only. Okay, so what does this mean? Filter out of the blood. It's referred to because, right, glomerulus, right, they are actually receive the blood from which side? It's the afferent arteriole. Okay, it's the afferent arteriole. Afferent arteriole. Okay, this one afferent arteriole. So what will happen is they have a larger diameter. A large diameter within this arteriole. And then what will happen is this large diameter, right, they can force a lot of blood enter to the glomerulus. Okay, but not all substance can be filtered out of the glomerulus and enter to the Bowman capsule. So this space will be the Bowman capsule here. This empty space. Because later, right, the component which enter to the glomerulus, right, they need to undergo ultrafiltration, so they will being forced out. Okay, and get into the Bowman capsule. So they're asking which substance they can be filtered out. Filtered out like this, or see, they're actually getting out from the glomerulus and enter to the Bowman capsule. So that means that the substance that which is required very small particle size. So A and B will not be answered because it includes proteins. Protein is considered a very large molecule. Okay, and that's why protein is considered a very large molecule. So proteins cannot be filtered out from this glomerulus get into the Bowman capsule because it's too large already. Not only protein, any kind of substance also cannot be filtered out. proteins, red blood cell, and the platelets, right? These three substances. Okay, so they cannot filter out. So it includes what? Proteins, red blood cell, platelet. So other smaller substances, right? Glucose, salts, urea, water, they can get into Bowman capsule. And then throughout this process, right, they will get into the proximal convoluted tubule and get the choline ducts. So they will do the reabsorption and also secretions of the waste product. Yeah, so the answer is number C. Okay, are you clear? Number C. Filter out means that they will get from the glomerulus, get into the Bowman capsule. So only small particle can be filtered out, get into the Bowman capsule. And then for this one, this is number is A. Okay, because renal artery. Okay, what's mean by renal artery? Renal artery is they actually transport the blood from which organs and get into the kidney. Where you get the renal artery from? That's where you can get the renal artery. Renal artery is originated from which? From which organs? Hearts. Still remember? Hearts. Okay, hearts. 你的心脏, your heart. Hearts. So you just imagine that this is the heart, right? I just put a love. Okay, means it's heart. Your heart, and then it consists of what? They have a very large blood vessel, help to pump the blood out of the heart and get to the body tissue, right? So this is with the aorta. So remember the aorta. So the aorta, right? After they being branched out from the heart, right? Actually, it's being divided, okay, into the smaller artery branch. For example, renal artery is one of the branch which originated from the aorta. So A will be the aorta, it's not vein. And then, yeah, for this one. Okay, since that you already finished your exercise, right? First, you carry the exercise and then you didn't drink water. So what happened to your urine concentrations after you just end the exercise? So that means that urine concentration will be increased, but the volume will be decreased. Do you know why? Because you didn't drink any water, right? So your body will become very dehydrated. So that's why your body will use up the waters, okay, to do their metabolic process. And then you did not drink extra water, right? So that's why it will cause your body become very dehydrated. Okay, so that's why your urine volume will decrease. The reason that urine volume is being decreased is because that you didn't drink water, right? So that's why you produce a very high concentrated urine. No water drink will produce a high concentrated urine. Okay, the reason they will produce this high concentrated urine is because that the body want to retain the moisture. In the body. So they will reduce the excessive water secretions from the body because they want to conserve water. Since you don't have new water input, right? So that's why your body will try to conserve the water in the body. So they will reduce their volume. So when they reduce their volume, right, they will cause increase in the urine concentrations. So what will happen if you drink too much water? And also, what will happen if you drink too much water? What happened to your urine volume? And as well, if you drink too much water, what will happen to your urine volume? Increase or decrease? It's increased, right? It's increased. Volume will increase. And how about concentrations? When the volume of yes, it's decreased. Yeah, it's inversely proportional. Yeah, if you increase your volume, right, that means the concentrations will be reducing, decreasing. And then another one. Okay, for this, both of you get wrong answer. Why the answer is donkey? Which part contain the higher concentration of proteins? Do you know why A is not answer? It's because that proteins is large particle, right? It's large. If it's a large particle, right, how they can be in transport from the glomerulus get into Bowman capsule? They cannot, right? Because it's too large already. So they will not get into the part A, because they can't even undergo ultrafiltrations to get into the Bowman capsule. So that's why if there's a protein inside the blood, right, after they will bypass the glomerulus, right, they will still left protein inside at here. Because because of protein, they cannot pass through this small hole of the glomerulus capillary and get into the Bowman capsule. That's why there will still be protein at here. Okay, at this side. And then why at this side, right, they have a higher concentration of proteins compared to other region is because that when the blood, okay, initially the blood, they have what? Initially the blood, they have the proteins. Not only proteins, they also have what? They have the proteins, for example. The incoming blood, they just get entry into the glomerulus. Incoming fresh blood. What do they have? They have the protein. Red blood cell. Red blood. Okay, and then mineral salt, urea, glucose. So what happened is the smaller particle, which is salts, urea, glucose, can get into the Bowman capsule. But the larger particle, right, which is a protein, red blood cell, platelets, right, cannot get. Okay, they cannot get into the Bowman capsule. So that's why that they will still being excreted out at here. And then since there's no presence of the mineral salt and the urea and glucose, right, so that's why it made the concentrations of the proteins and also the red blood cell, right, become very high. You get what I mean? So that is donkey. Just remember, if the particle too large, too large in the size, like the proteins, red blood cell, and platelet, you can see you have these three elements which is very large particle size, and then they cannot diffuse through the glomerulus. What happened to that? Yeah, I hope is, you won't feel very difficult if answering those questions. You just need more practice also, okay? And then after you do that question, right, you need to understand. You need to understand the theory before you do the questions. It's very important for you to understand the theory first. So in the healthy people, which substance is being completely reabsorbed into the blood from the kidney tubule? So why you say urea? Urea will be reabsorbed into blood from the kidney tubule? Not really, it's glucose. Urea at the end, right, they will be excreted out in the urine. So there will be no urea reabsorbed back into the blood. Understand? But they will be other neutral molecule like the glucose. Glucose, after they get into the Bowman capsule, they get into proximal convoluted tubule. So from the tubule, right, they will get back to the blood. Just like this. Just imagine like this. Okay, and then here is the Bowman capsule. Sorry, do I record? Let me check. I'm recording, right? Yeah, I'm recording. Okay. Yeah, I'm recording. Yeah, so here is the Bowman capsule. See, here is the Bowman capsule, so something like this, right? And then continuation to the proximal convoluted tubule here. Okay. See, here is the proximal convoluted tubule, and then get into the Coriolis duct. So here is a PCT, okay, PCT. And here is the Bowman capsule. Bowman capsule. So which substance is being completely reabsorbed into the blood from the kidney tubule? So, okay, just imagine, right? What they can get an entry into this Bowman capsule space is a small particle. And then it includes the glucose, salt, urea, and the water. It includes in those four substances. Now they will get into the Bowman capsule. So the other three substances unable to get into into this Bowman capsule is what? Is the red blood cell, platelets, and also the red blood cell platelet. And also a plasma protein, okay, and also a protein. Just these three substances too large in particle size, and then they cannot diffuse inside. So what will happen is the glucose. So now you can see, right? Glucose, urea, water, salt, they can enter the Bowman capsule. So after they enter the Bowman capsule, right, they will go to where? They will go to the PCT, which is the proximal convoluted tubule. Proximal convoluted tubule. Okay, so this PCT, right, what they do is they will do the reabsorptions. They will do the we call it selective reabsorption, choosing the substance to be reabsorbed. Okay, they are trying to choose the substance they will be reabsorbed back. So just figure out like this, right? This is the tubule, okay? Like this. And then here, back to the coil state, and then get into the coiling duct. Okay, so here is a DCT, distal convoluted tubule. Okay, distal convoluted tubule. And then here will be the coiling duct here. So after the urea, mineral ions, glucose, they get into proximal convoluted tubule, right? So along this proximal convoluted tubule, there will be a network of blood vessel surrounding it. Okay, there will be a network of blood vessels surrounding this. Surrounded this proximal convoluted tubule. So what happened is the proximal convoluted tubule, right? They are doing what? They are doing selective reabsorptions. So they are trying to reabsorb. What's mean reabsorb? Reabsorb means that the movement of the substance from the tubule get into the blood capillary, which is surrounding it. Okay, so means that the substance from the tubule and then get into the blood vessel. So now the glucose. So that's why at this stage, right, glucose will be 100% being reabsorbed by the proximal by the blood capillary from the proximal convoluted tubule. So just imagine that glucose will be reabsorbed back. And then there will be 100% of glucose being reabsorbed from the kidney tubule get into the blood vessel. From the kidney tubule. Okay, understand? So that's why the urines contain glucose or not. The answer is no, right? If you're able to test the glucose in the urine, that means there will be some issue happen to your kidney. Okay, maybe your kidney is undergo inflammation at any certain spot, so it will cause the failure of the reabsorption of the nutrient molecule get into the blood capillary. So the answer is A, glucose. Because glucose is the only one that will 100%, right, reabsorb at the proximal convoluted tubule. Just remember that. Just remember that proximal convoluted tubule, they will absorb the glucose. And then for this one, what is filtered out of the blood in the glomerulus to the kidney tubule? So the answer is A. Okay. Why C is not answered? Because that, you see, from the glomerulus, right, you can get into where? You can get into the proximal convoluted tubule. So both glucose and urea, both of them, they can also get into the proximal convoluted tubule, which is the kidney tubule. Okay, any kind of questions? And then for this one, which row is correct? Higher concentration of urea in the blood once they enter into kidney. Then become lower concentration of urea in the blood entering the liver. Okay, what the answer is not A? Sorry. What the answer is B? What the answer is B? It's not A. It's because that you just imagine that how the blood they will get into the liver and then to the kidney. And then which row show that is correct. So if you have too much urea in the blood, right, the next step you should do is you should get into the kidney. So kidney will try their best to help to do what? They help to do the excretion of the urea. They help to do the deamination. Okay, deaminations. Then this one, lower concentration of urea in the blood, right? So if the blood, okay, if the blood from the outside get into the liver, okay, like just like this, okay, entering and leaving the liver and the kidney, right? So when they enter to the liver, right, it's still low concentrations. When they're leaving the liver, right, there will be what? High concentration, right? Because liver is the organs which they will produce the urea. Okay, they will become very high concentration after they leave the liver. Maybe I write out here. So the answer is B. Leaving the liver will be very high concentrations. Before they enter the liver, right, there will be very low urea in the blood. So the liver is playing out their function, help to clear those urea to become less toxic. Okay, and then any kind of question I haven't discussed with you. Okay, another for this one. Which role match the function to the correct organs? So does this 1, 3, 2. The one who excrete carbon dioxide is referred to which one? Lung. Excrete the urea is referred to the produce the urea is referred to the lung. Excrete the urea is the number 3, kidney. Okay, and then this one, mammalian kidney. So number S, right, will be the cortex. The most outermost layer, okay? The outermost layer will be the cortex. Then Z will be the medulla. Okay, see, medulla is inside at here. Medulla is at here. Then Y will be the ureter, okay? It's not urethra, but it's a ureter. Ureter later they will connect it to the organ. And then any kind of other questions. Okay, and then for this one. This is actually what they do is they show you the concentrations of the blood plasma. And then sodium ion in the blood plasma in the glomerular filtrates. They pass through the glomerulus and the urine. So what is the percent of concentration of sodium ion is reabsorbed by the kidney tubule? So that is 9.9, okay? How they can get 9.9 is because that, you see, glomerulus filtrate, right, we still have 141 sodium ion, but after urine, 127 already. So 127, maybe I can just make something like this. Okay, just imagine, yeah. What is 141? What is 127? 14 unit. Okay, 14 unit over 141 times times 100. Okay, 14s over 141 times 100. So that is 19. I mean this 9.9. Okay, because you use this one 147 to divide by 127, right? Okay, the answer is 9.9. Can we get this? Because, right, you can see after the sodium ion, they pass through in the liver, they will only absorb about 9.9%. And then this one, which statement is urea is corrected? Okay, A, B, C, D. Urea is made in the kidney and then they will remove from the body by the liver. So this one is urea is made in the kidney and then removed from the body by the liver. Is this answer correct? No, right? So the answer is B. Okay, this one B. So urea travel from the liver cell because the liver cell they will produce the urea. So they will get the kidney to be excreted out. Yeah, that's all. And then for this one, yeah. Okay, just remember that what happens if you have a very low glucose concentrations? So if you have very low glucose concentration, right, your body will try hard to increase the blood glucose level back. So the body will try to break down the glycogens, okay, stored inside the body by the glucagon to convert glycogen become the glucose. And then the substance excreted out from the human body include those of carbon dioxide, urea, and water. Those are the substances. Okay, then what is the response to the low concentration of glucose in the blood? So the answer is B. If you have a very low concentration of glucose in the blood, right, you will have the hypoglycemia. Hypoglycemia refers to 低血糖, okay? So you feel like lethargy and then you feel like fainting. Yeah. So that's why the answer is B. Okay, and then for this one, what is the response to the low concentrations of the glucose in the blood? Glucagons will cause the body to convert glycogen into glucose. Yeah. So if you have very low glucose in the blood, right? That's why your body will try hard to increase back the glucose. So that's why they will try to convert the glycogen stored inside liver muscle become glucose. So that's why you can help to increase your blood glucose level back. Okay, we finished discussion already. I hope you have more understanding toward this topic. Yeah. Just remember that liver produce urea, right? Liver produce urea. So urea will be excreted by the kidney into the urine. So far, so good.
Yep.
Okay. Please don't repeat the question. Don't repeat the mistake again. You're going to have your exam another 7 more months. Okay. Times fly very fast. You are going to have exam at next year, May, yeah. June or May. I hope you have sufficient time to prepare for it. And then for this one, chapter 15, last time I haven't discussed with you. So I also have marked already. And then for this one, the number of bacteria, right, which is resistant to antibiotic is increasing. So what has contributed to this situation? So under is B. It's not because of A. It's because that usually the patient, they do not finish their course of the antibiotic. Okay, their doctor asked them to finish all, but they didn't finish. They just eat few only. Okay, and then they will get better. They recover already. So that's why they feel that they didn't need antibiotic anymore. Okay, and then this one, which statements about microglial? Yeah, that's a spoil. And then for this one, which one? I want to show some mistake. This one. What happened to the antibiotic is used to kill the bacterial infections. So the answer is this one. 4132. Okay, starting from number 4. So you have antibiotic kill most bacteria, right? So the antibody resistant to survive. Okay, and then the antibody resistant bacteria will produce. They need to survive first before they can reproduce, right? At least they need to survive to the surrounding. They can adapt to surrounding, and then they will start to produce. And then for this one, which substance in the tobacco smoke directly reduce the oxygenation of blood? So that is carbon monoxide. Okay, I'm not sure you still remember or not. Last time we talked about this one, the drug. Okay, if you compare to the hemoglobin, right, and this one, the nicotines. And also during smoking, right, they also will release the carbon monoxide. So this carbon monoxide, they will bind irreversibly to the hemoglobin. So they will reduce the capacity of the blood to carry the carry the oxygen. Means that if the hemoglobin itself, right, okay, initially, right, they will bind to oxygen, but now there's no oxygen, they will become HbCO. Okay, they will become this one, HbCO. I'm sorry, HbO2. So since that you have the carbon monoxide attached with it, so they'll form a strong bond with the carbon monoxide, with this one, carbon monoxide. Right. So you actually reduce the capacity to carry the oxygen. So what happened to the carbon monoxide? Also, it will cause the narrowing of our blood vessel, okay? Or our respiratory airway, sorry. It's about our respiratory airway. Then go back to the questions. So it's carbon monoxide. Carbon monoxide, they will bind irreversibly to the to the hemoglobin. They can just bind to hemoglobin. And also the number of hemoglobin is being limited. So this will reduce the space of oxygen bind to the hemoglobin. And then for this one, describe the use of antibiotic. So example is 1 and 4. You use antibiotic, you want to prevent infection only. Okay, so which practice will increase the chance of development of antibiotic resistance in the bacteria? Number 1 and number 4. Means that you just use antibiotic without any control. Just use antibiotic without any control. So you can see at number 1, using an antibiotic to prevent infections. Number 4, using antibiotic to treat live infections. Okay, here. Any other mistake? Let me check. It is here. Okay, this one showing what? This one showing that this is a graph. So what answer number 2 and number 3? So you can see that smoking cigarette for more year increase of the risk of the lung cancer. So you can see, right, when you smoke more, the lung cancer also increase more. So smoking cigarette for many years increase the risk of the lung cancer. Yeah, they do increase the risk of the lung cancer. So that is number 1 and number 2. Okay, this one, smoking more than 20 cigarettes per day for more than 50 years has a higher risk of lung cancer. It did not mention about this one. The question did not mention about this detail. So answer is number 1 and 2. Sorry. Yeah. Okay. So yeah, my document is moving now. Okay, and then other type of questions haven't discussed yet for this one. The disease cannot treat with the antibiotic. So it's number 1 and number 2. It's not because that the patient resistant to antibiotic. Okay, it's just because that it's not bacterial disease or it's a virus. If it's a virus disease, right, we cannot treat them with antibiotic. Antibiotic will not kill virus. Do you know why? It's because that virus structure, right? Do they have cell wall or not? Is there cell wall inside the virus? Okay, finish already.
No.
And this one, is there a cell wall inside the virus? No, right? There's no cell wall. Okay, so last week, right, we stopped until HIV. So today we'll continue to discuss about the HIV. We'll finish it. And then we will discuss about the paper 4. Okay, paper 4. Most important is the paper 4 question. And then you can finish your paper 2 again. Let me check where is the HIV. Okay. We go back to the HIV first. Okay. So HIV is an immunodeficiency disease. Means that this kind of disease, right, they will help to require lower down your, it's not lower down, I mean it's weaken. They will help to weaken your immune system. So how they can weaken the immune system? Hi, are you still there? Okay. What happened to my laptop? You can see me, right?
Yeah.
Oh, okay. I have some issue with my laptop. So what happened to your immune system if you get infected by HIV virus? HIV virus will directly destroy the lymphocyte, right? Destroy the lymphocyte. Lymphocyte is a white blood cell. And then the function of lymphocyte, which is a white blood cell, they will help you to defense. They'll help you to defense against the pathogens, right? So now the virus, they will directly kill the lymphocyte. They kill the lymphocyte. They destroy the lymphocyte. Okay. They not only destroy one lymphocyte, but they will destroy all of the lymphocyte. Thousands, hundreds of lymphocytes. So make the lymphocyte, right, become a machinery, see, to multiply, to produce their viral copy. So initially, you only have one virus, in fact, one lymphocyte, right? At the end, you'll be about the lymphocyte, they will rupture. They will rupture, they will release about thousands, 100 copy of the virus. So like this. Okay, so means that this HIV virus, their structure. We talk about the virus structure, right? Virus structures, they have what? Like this, okay? Capsid protein codes. Capsid, or known as protein codes. So inside this protein code, right, they have the nucleic acid. And then they are being surrounded by the membrane. Okay. This is the membrane surrounded. And then the surface of membrane, right, you have what? You have this one. Like this, see, this one. What is this? This is we call as a glycoproteins, or we know as surface proteins. Glycoproteins. Or we call it surface protein. So this glycoprotein, right, they have their own structure and shape. So if the virus, they want to get into the host cell, so this is the host cell. Host cell. So if the virus, they will get into the host cell, right, they will infect the host cell. So the host cell, their surface, right, they also have their own receptor. See? So in this host cell will belong to the lymphocyte. Lymphocyte is a host cell. So on the surface of the lymphocyte, they have the receptor protein. So the virus, okay, they will bind to the receptor proteins. The glycoproteins, okay, the glycoprotein of the virus itself, right, they can match with the structure of the receptor proteins. So that's why they can just get an entry into the lymphocyte. So after they enter into lymphocyte, right, they will just inject its genetic material, which is their DNA, into the lymphocyte. So, and then they will get in the genome, which is they will enter to the nucleus. So HIV will use the lymphocyte as a machinery to make a copy of the virus itself. So at the end, right, you have made a lot of copy of virus inside this lymphocyte, so they will burst. So the result and the outcome of the HIV infection is they will cause the rapid reductions of the lymphocyte. So if there is not much lymphocyte exists in the body, right, there will be a rapid decrease in the production of the antibody. If you don't have enough antibody, right, so there's no there's no more antibody will help to bind to the virus or the pathogens. So that's why you are easy to get infected by any kind of pathogens, okay? Even a simple flu or cough, right, they have ability to kill you because now your immune system is very weak already. Any of the virus, any of pathogens, right, they have a possibility to kill you because you don't have any lymphocyte to help to protect you. You don't have lymphocyte, you don't have antibody, you don't have the antibody to help to kill the pathogens. So that's why any pathogen, they can kill you if you have this one immunodeficiency. So usually HIV infected virus, right, it's not die because of HIV. It's die because of other disease. Okay, other disease like the cough, sneezing, or the pneumonia. Okay, you know, pneumonia, 肺痨, pneumonia. So pneumonia also, they can be like the outcome result of the weakened immune system. Because you don't have any immune response already. So and then how you're able to control the spread of STI means that the STI refer to sexual transmitted disease. So limited number of sexual partners, or you should have a protected sex by using the condoms, and then you should be getting tests if unprotected sex or sex with a multiple partner has occurred. Okay, and then raising awareness by the education program. So here's a way that you can control or you can protect yourself from getting this HIV infections. Okay, because once you get infected with HIV, right, there will be a lifelong infections. You only can eat the medicines to help to control the infection only. You cannot totally recover from the HIV infections. Okay, so now we can discuss about the chapter 16, right? You can open these notes. Where is it? Here. Not here. Last time, waiting for a while. This one, okay. You can open this note. This one. Chapter 16, paper 4 with the answer. You can open this one. Okay. And then we can discuss some of the questions. I just want to show you that how we write the I mean the answer during exam. I mean, how can we construct the sentence? So that's why when the marker they mark the answer, right, they can clearly see your point and then they can give you mark. Okay, so let's go through some questions. Okay. We talk about HIV first. Okay, just now we talk about HIV, right? Okay, disadvantage of the sexual reproductions. So what is the disadvantage? So their advantage is they will, I mean, how to increase the genetic variations. So this increased genetic variation, right, will give, we call it selective, because like something like they have an ability to produce a better, stronger offspring if you do the sexual reproductions by increasing the genetic variations. But the disadvantage of this increased genetic variation is because that the offspring they're being produced, right, they will not totally inherit the parents' traits. Okay, maybe you can able to produce an offspring which is they look. I mean, when you look at the external structure, right, they look a bit different from their parents. So it's because that if you are doing the sexual reproductions, the offspring will not totally 100% inherit the trait of their parent plants. Another thing is the disadvantage of the sexual reproduction is you are requiring more time and more energies to do the mating. So it will be time-consuming process to achieve a pure breeding stock. Okay. So you should know how to describe the process of meiosis. Meiosis is a kind of nucleus division. So you produce these 4 haploid cells. Okay, and then this one you will discuss in next chapter. It's about how to draw a genetic diagram. And then for this one, you can read about cell. Okay, for this one, the role of lymphocyte. Okay, you can write down that lymphocyte, they will help to fight against the pathogens or infections by produce the antibody. Okay, they'll produce the antibody which is specific to invading or kill the certain pathogens. So remember that our immune system, right, actually is being depends on the active immunity and the passive immunity, but majority will be the active immunity, because active immunity is the immune response which is acquired by our own body. Okay, so what happened is the HIV infections, they will destroy this active immunity. Okay, and then this one they talk about HIV. Okay, HIV, they invade a specific lymphocyte, they coordinate the immune response. So this change the number of the lymphocyte following the infections, they have not been treated. So you can see, right, over the time by times, lymphocyte, they will be decreasing. They're actually decreasing. So describe the change in lymphocyte number following the HIV infections. So you can say that how you can describe the change. You can see, right, they are actually decreasing rapidly. So you can say that there is a steep decrease over the time. Okay, over the first 2 years, dropping out about 500 cell per millimeter cube. Okay, so they're actually decreasing from here, which is about 800 to 500 at here. Steep decrease like this. And then this is followed by the slight recovery, with a count increasing to about 660 per millimeter cube. And then there's a gradually but steadily decline. Okay, so you can describe like this. Gradually, because they decline very slow, like not too fast. So gradually. Gradually means what? Means that gradual means slow. There's a very slow decreasing trend of the lymphocyte over the years. So we can write gradually but steadily decline until they fall into very low level. So it's about 14. Here's about 14. So, and also you need to calculate about the skew number. So here will be because there will be 5 skew, right? 1, 2, 3, 4, 5. So 200 divided by 5 skew will be how much? 40. 1 skew will be 40, right? 40. 80. 120. 160. So when you drop until here, it's about 40. If they ask you to describe the change or trends of the graph, right, you need to describe like increase, decrease, and then you need to describe the trends. Increase rapidly, decrease steadily, no change, or maintain at a constant rate, like this. And then you need to add on the value figure inside if you can. Just copy the unit at here. If you put the value right, please remember to put the unit as well. And then describe the effect of the body result as untreated HIV infections. So they will destroy the lymphocyte. So this will reduce the ability to produce an antibody and mount an effective immune response. So the person, right, they will become extremely vulnerable to the opportunistic infections. See, like the pneumonia and tuberculosis. Because you don't have lymphocyte to help to produce the antibody. So antibody cannot eat the pathogen already. So you'll be dying off due to the opportunistic infections, which is we call as secondary bacterial infections. So this is known as what? This is known as acquired immune deficiency syndromes, can cause a weight loss and reduce lifespan. Okay, this is IVF. Okay, last time we discussed already. Okay, for this one, IVF. So what kind of injections there will be performed for the IVF procedure? So they will inject the hormones. See, the hormones they will be injected to stimulate the egg cell development is the follicle stimulating hormones. Okay, because they need to encourage, they will encourage the ovary to produce more mature ovum. So that's why they need to inject this follicle stimulating hormones. So state when during the menstrual cycle, the hormone should be injected. So they will inject it at the starting, at the starting of menstrual cycle, which is between day 1 and day 10. Usually usually you will inject in first 5 days enough for you already. Okay, first 5 days. In the first week. Then how about the string C? So you can see, right, the string C, what they do is they will use needles, okay, and puncture the ovum. So they will suck out one of the ovum. Yeah. So the string C actually is trying to remove the egg cell from the ovary. Okay, they just remove the egg cell from the ovary. So this one, an X. An X should be drawn on the lining of uterus to indicate where the embryo are placed for the implantations. Okay, so where you can draw the lining of uterus, you can draw around here. So here is the lining of uterus. Okay, the social implication of IVF, which is they refer to the outcome. So what will be the outcome? So it will allow sorry, it will allow the infertile couple. Infertile couple means that this couple, right, they have something like the issue with their reproductive activity. For example, in the male, right, usually the common issue they will have is their sperm is not healthy, not a healthy sperm. Okay, means that the sperm is not fertile. Maybe the sperm is not immature. I mean it's not mature enough. So that's why the sperm, right, unable to fertilize the ovum because they have a very poor sperm quality in the male. So this will make them become infertile. So IVF, it can help them, the infertile couples, to have their own children. Okay, or single parents means that in the future, right, the women, they can get pregnant without any male, because what they can do is they can buy the sperms from the sperm bank or something like that, fertilize with their own ovum, or the same-sex couple to have a children. So the procedure, right, is very expensive and may not always be successful. Okay, maybe you can curious about how is the successful rate for the IVF. Successful rate of the IVF is about 40 to 65 per treatment. Okay, sometimes it can be about 80%. So it's not about 100%. You still can failure. You still can have failure of cannot produce any baby. So not always being successful. So it also raised the articles and the religious concerns for some people. So that's why the IVF, they can lead to the multiple birth and the possibility of genetic screening of the embryo. Okay, do you know why IVF can have this multiple birth? Is because that in every cycle of the IVF, right, you can take out maybe several ovum and several sperm. So that's why you can produce about more than one embryo. So you can have multiple birth. So the advantage is they can do the genetic screening of embryo, means that you can do the gradings. You can do the embryo gradings to choose a best embryo to implant into the uterus. So these are social implications. Okay, so the advantage of the advantage of this sexual reproductions is, for this one, genetically identical radish. Genetically identical is referred to the mitosis. Okay, mitosis. So mitosis, right? They will do what? They will reduce the genetic variations. Just remember, meiosis will increase genetic variations, but mitosis will reduce genetic variation. So to ensure that any difference observed in the growth of plants are due to the different mineral iron solutions they are grown in, and not to inherited traits. So this will result more reliable between the groups. Okay, so this is their advantage. If you produce a totally same offsprings, you actually reduce the genetic variation, so you can have preserve their parent traits. Okay, you can preserve the traits from the parent itself. And then this one, yeah. Let me discuss with other kind of question. Okay, this one you can have a read. I think it's very good reference for you because you can read and then you can know how to answer by using the correct keyword. Then for this one, yeah, okay, the sexual reproduction in the flower, right? And then you already know already. Okay, so let me show you other kind of question, which is, yeah, for this one, see, contraceptive methods. Okay, so why we should use these contraceptive methods? So we use a contraceptive method by how many type of methods? Chemical ways, physical barriers, and using this one. So like the condoms, okay, or you can say the femidums, they can prevent the transmission of a sexually transmitted infections. Okay, so the purpose is they want to reduce the transmission of the sexual transmitted infection only. That's all. So this is their this is their purpose. Okay. So this is their method. Okay, methods. Use any methods: condoms, contraceptive pills, or other chemical method. So usually the condoms and femidums, right, will be common method in the birth control because it's quite convenient. Okay. Then stage 2 hormones used is the estrogen and progesterone. Just remember that contraceptive pill, right, they only have these 2 hormones inside only. Okay. Then during the IVF, right, starting of IVF, they will inject the follicle stimulating hormones. Because if you have a very high amount of estrogen and progesterone, right, so what we do is actually you will inhibit, you will able to inhibit on the development of the new follicle. Because estrogen and progesterone, right, they are focused on increasing the thickness of the endometrial lining. Okay, why contraceptive pills do not contain FSH? Because FSH will stimulate the growth and development of follicle. So we don't want the follicle to mature in the ovary because it will increase the chance of the ovulation pregnancy. That's why we want estrogen and progesterone only. Because these 2 hormones, right, they will inhibit, okay, they will prevent any ovulation and pregnancy happen. So other chemical methods, which include, okay, spermicide. You can see, right, this spermicide. Spermicide is a kind of chemical substance that can help to kill the sperm. And other, okay, you can use a patch or injections, which is referred to. You can try any other ways to inject your body with the progesterones and the estrogens. Okay, and then for this one, the percentage of last 3 bars added together is 90%. So suggest why the percentage of women who use any kind of contraceptive pills, so contraceptive methods, is not equal to the sum of the last 3 bar. Okay, it's because that they might use different type of the contraceptive methods. Okay, it's not only, I mean, they not only rely on condoms or contraceptive pill only, but they still got use other method. Then, okay, any kind of questions we haven't discussed. In next chapter, right, we'll talk about the haploid and diploid number of chromosomes. So you can see, right? So the importance of gametes to be haploid is because that the haploid gametes, right, they will need to fertilize together to become the diploid zygote. So they can restore back their diploid number of chromosomes. And also, this is to prevent the chromosome number from being doubled up at fertilization. So just imagine that if your sex gamete didn't separate, right, if there are still 46, okay, in the sex gametes. For example, like in the ovum and sperm, if there are still 46, right? So what will happen is, if this sperm, they fuse with another ovum, so this sperm is 46, it's because they are not being divided yet. And then this is 23, okay? This is a normal ovum. So 46 plus 33 is about how many? 69, I think. So this 69, is this chromosome able to exist in our cell? No. The answer is no. That's why they must be undergo meiosis to separate their chromosome number, to halve their chromosome number, become 23. Because they want to restore back, which is 46 later. And then they want to ensure this constant number of the diploid, or of this chromosome number, which is 46, passing from generation to another generation. And then this one we'll talk about this later. Okay, what's the mutations? This is in the next chapter already. Yeah, okay. And then for this one, okay. This is an interesting question. See, glycogen is a storage carbohydrate in the animals. It's made from the glucose itself. Okay, so you know that glycogen is a polysaccharide, right? It's just same to the starch and cellulose is a polysaccharide. Glucose is a monosaccharide, okay? It's a monosaccharide. So why this cell have many mitochondria? It's because that if you have a lot of mitochondria in the cell, right? So what will happen is the mitochondria, they will do a lot of cellular aerobic respirations. Okay, so that means that if the cell, they will convert the glucose to glycogens, right? They require a lot of energy, help them to do this process. So that's why they require a lot of mitochondria, okay, help to release the energy to supply to the cell. So they have more energy, they can do this process, which is they can convert this glucose become the glycogens. Yeah, so any kind of the molecule catalyzed reaction. So you can see these keywords, catalyzed reactions. It refers to what? Enzyme. Okay, refer to enzyme, catalyzed reactions. Okay, so for this one, how fetus able to obtain the glucose? The fetus obtain the glucose from its mothers, okay, by the simple diffusions. Okay, so remember simple diffusions, which is from the mother bloods, right? They can get to the fetus through the placenta. They need to cross the placenta, okay, from the high concentrations in the mother bloods. So just imagine that mother blood, right? They have very high concentrations of the glucose and the oxygens, right? And then they get to fetus bloods. Fetus bloods, they have a lower concentration of glucose and oxygens. So they travel to the fetus through the umbilical cord. Okay, just remember that umbilical cord, right, is actually composed of the artery and the vein. It's composed of this artery and the vein, umbilical cord. And then for this one, yeah. Okay, see, calculate the percentage increase in the glycogen concentrations in the fetus between day 10 and birth. Okay, so you just imagine that day 10, right? Here, how they able to increase their glucose from day 10 increase until here. So you need to like do some simple calculation. Okay, so day 10 is about 220. Okay, this one you can calculate by yourself. 220. Here will be 220. Here will be 600 above. Okay, so just and then over, you should over this the initial amount. So that's why you're able to find that you increase about 182%. So the answer is 182%. Okay, means that the amount of increase, right, you divide by the initial number, which is 220 times 100. So you get this one. And then you should able to describe the change in the glycogen concentrations. So how you able to describe the change? So you can say that initially, right, the glycogen concentrations from the day 10 until day 20 will be slightly reducing. It's not a steep reduce, but it's just like slightly reducing. But when the day 20 above, right, there'll be a steep increase. See, steep increase. So this is where you can explain. Glycogen concentrations, they remain low and stable at around 220 until day 26. Then after that, they begin to increase steadily, peaking about 620. So then usually, right, this kind of question, they will give about 6 marks. Then you should write about the trends. Okay, so now you already described about trends. Trend is you say that remain low, okay, stable. And then after that, you can say increase steadily, or you can say that there's steep increase until the peaking. Peaking, they reach a peak on about how many? About between 60 to 70 days, which is 620. And please include the figure as well. So this is being stimulated by the hormones insulin which cause the glucose to be converted into the glycogens, right? So see, now the glucose, right, is being converted to glycogens. Yeah. So immediately after birth, right? Immediately after birth, the concentrations concentrations drop sharply to around 340. So as the hormone glucagons, they stimulate the breakdown of this this store glycogens back into the glucose to provide energy from the newborns. So what does this mean? This means that you can see, right, the glycogens initially is increasing, but after that it will be reducing. Okay, so when the glycogens they reduce from 620 until 340 like that, 340. So actually this is a place where the glycogen is being break down. Okay, so this is a site where the breakdown of the glycogen. Okay, glycogen is being break down, become what? Glucose already.
Yeah.
Then how about here? Here to here, what will happen? There's an increasing in the glycogen concentrations. So why the glycogen concentration increase is because the insulin effect, okay, means that all the glucose, right, is being converted become glycogen. So that's why they will be increasing in glycogen concentrations. So you should say that from day 20 to about day, how many days? Here will be about day 66, something like that. Okay, please include the figure as well. Yeah. So you can see that the increase in the glycogen concentrations is due to the hormone insulin. So all the glucose is being converted to glycogens for the storage. But after birth, right, the glycogen drop very fast to about 340 per gram. Okay, which is here. 340. 340. Right? So you should say why? Because that the hormone glucagons, okay, now they are being take over the roles. Before that is insulin, right? Insulin, they will try to convert all the glucose become glycogen. So now when the glucagon take over the roles, right, they will convert all the glycogen become the glucose. So they will break down the stored glycogen inside the livers and to the glucose. So because they need to provide energy for the newborns. Yeah. So this is describe the change, right? And then explain how. Describe and then explain. So these first two or three marks, right, they will be given if you're able to describe the change, okay, the change of the curve. Then this one, right, you'll be able to describe the reasons, the reason behind, the reason happened behind the change in the graph. Why is it increase? Why it decrease? Why it maintain a constant? Why it's picking up? So it's about 6 marks, okay? So when you write about this describe, right, please write as detailed as possible. Because describe, you refer to the something like essay. It's kind of essay. Okay, this advantage of breastfeeding is sometimes maybe if the mother, right, for example, if the mother is being infected by HIV, you know, so this HIV virus, right, they can pass through the placenta to the to the baby also. Or sometimes if their milk, right, they have the harmful substance, right, something like drug or pathogens, so they can pass from the mother to the baby through the milk. Okay, so also it's time consuming and tiring for the mother. Yeah. Okay, for this one, I just discussed about the some question which is quite important, and then they will help in your understanding. Yeah. Okay. Any kind of questions? Okay, for this one, yeah. Okay, just have a read on the reproductive structure of the male and female. And then, okay, what do we mean by species? Species is to refer a group of organisms that can reproduce to produce a fertile offspring. Okay, so they must be same species. If same species, right, that means that both two words need to be same. Means that Homo sapien only can make to Homo sapien. Like this. Two words, right? Two words. The genus and the species name, they must be exactly same to each other. So you can see, right, this is the reproductions in the potato plants. So this is a kind of we call as a asexual reproduction. So how they can do this? Okay, they just cut the tuber, right, for the potato and then plant on the pot, and then they'll grow into new offspring already, new plantets. So this method of reproduction, you should say that is the asexual reproduction. Okay, so what is the advantage? Advantage is that it's faster, so they can produce a lot of plants in a very short time, and then they produce a genetically identical to the parents. So means that if the parents, right, they have the desirable traits, right, they can directly inherited by the offsprings. Yeah, for example, like height yield and the disease resistance. Okay, so this is the advantage. And also, this kind of the asexual reproduction, right, is you can save a lot of resource. Not like the sexual reproductions. You need to, you need more longer time, and it's an energy consuming process. So this one you can save more energy. It's much more easier to do this asexual reproductions compared to the sexual reproductions. Okay, and then this one, why plant store starch? Starch is a kind of energy storage compound. Remember about this one. So it's a kind of energy reserve. Okay, so why plant need to store the starch? It's because that when the plant unable to do the photosynthesis, right, at the night time or during the winter, okay, during the winter time, right, there will be very less amount of sunlight, or so that's why they might be entered to the dormancy stage. So that's why they still need to maintain their metabolic growth, right? So that's why now they will use the starch become the source of food. So previously they will do the photosynthesis, so they will get the glucose become source of food. So now they stop the photosynthesis already, so they just get the source of food from the store starch. From the store starch. Okay, and then for this one, the meiosis, right? I think we discovered this before. And then you should know how to write about how the fertilized egg, right? They can, how the sperm able to fertilize the egg cell. So you should write about the keyword. See, because zygote, and then they have the jelly cord hardens. Okay, so after they form the zygote, right, they'll be going down to the oviducts, and then they'll undergo the mitosis cell divisions, then become a ball of cell known as embryo, and then implantation on the uterus. So this is a keyword that the examiner going to have a look on the keyword. So they will give you the mark based on the keyword. Okay, and then this one. What's meant by cell pollinations? The consequence of the cell pollinations for the population of plants. So, do you know what's meant by cell pollination? Another one is we call it cross pollination. This one.
Yeah.
Cell pollinations. Yeah. Cell pollination is happen in same plant only. Okay, in the same flower of plant. So the advantage is they will lead to reduced genetic variations because they are just coming from only one parent. So this will increase the chance of the offsprings inherited a harmful recessive genetic disease. Okay, and then you also reduce their genetic variation, right? So you don't have chance to produce a better, stronger offspring. So you actually reduce the ability to adapt to the changing environment conditions. So this is a keyword that you can include in your answer. If you write something like you have a lesser genetic variations, right? So you have the reduced ability to adapt to the environment change. So if you're able to increase the genetic variation, right? So you have more ability. You actually increase the ability to adapt to the environment change. So you're able to increase the risk of disease because you have lesser ability to adapt the change. Okay. So I think other you can read by yourself. Yeah. So it's not too hard. Just you need to do more exercise and then know how to write the answer. Sometimes they will come out question like this. Just need to fill in the blank. So like this is very, very simple. Okay, so you know about sexual reproductions. And then any kind of questions, we haven't discussed it. This one. Ball or cell implant to the uterus after fertilization. So this is because embryo. Okay, sorry, embryo. Okay, embryo. This one embryo. And then compare the development of fetus in the early stage of pregnancy to its development in the late stage of pregnancy. So in the early stage of pregnancy, right, the focus of development is on increasing the complexity, okay, in major organs and body body system are formed. Then in the late stage of the pregnancy, right, the main focus shifts to rapid increase in the size and mass as the fetus grow and mature in preparation for the birth. So what does this mean? That it means that pregnancy, right? Okay, pregnancy in the human is about 9 months. Okay, 9 months. So the early stage is they refer to the first 3 months, 3 to 4 months. Okay, at this early stage of the pregnancy, right? So the embryo itself, okay, the embryo before they become a human baby, right? Become fully developed human baby, right? They're actually growing rapidly in their body system. Okay, so this is like you learn more if you study the higher level of biology. So it means that at this stage, right, they will form their organs. Like initially they'll form the nervous system first, and then they'll become the cardiovascular system. They'll form the hearts, right, the blood vessel, and then later they'll grow a muscle at their brain, something like that. They start to form their system. So at this early stage, right, there'll be the rapid growth, okay, and then this process, right, is very complex. And then during the late stage of pregnancy, right, late stage, right, means that maybe like last 2-3 months already. So the baby now is fully formed already. But now what they do is they just focus on increasing the size and mass. Means that they are actually increasing their number of the cell, or the size of the cell. So they become more mature or bigger. Okay, so this is just for you to have a look only, okay? So what does this mean by formation of organ? So it's like this. We call it organogenesis, okay? Organogenesis, yeah. During the embryology, okay? So it's something like this. Just imagine that after the diploid zygote is being formed, right? Diploid zygotes, okay, will implant on the uterus. So they will start to do the cell division, right? So the cell division, they will become what? They will become the morula, right? And then they become what? They become the blastula, something like that. So they will become into the embryo. Okay, they will become the embryo. So when they become the embryo, right? So now they become like this. They will form 3 layers, okay? So these 3 layers, they will develop into each of the organs, like the digestive organs, the respiratory organs, and also muscular organs, skeletal organ. So they become a fully developed baby, like this. See? So this is occur in the early stage of pregnancy. Okay, and then this one, what's the function of the amniotic fluid and the amniotic sacs? So the amniotic fluid, right, they contain several protective functions. So they will, because it's fluid, amniotic fluid is referred to the yang sui, okay? So they can help to protect the baby itself, cushioning the fetus from bump and shock. Because fluid, they can help to absorb the shock. So by this way, they can help to protect the fetus. So it can also maintain the constant temperature and allow the fetus to move more freely. So which is important for muscle and bone developments. So it can produce a sterile environments, protect the fetus from the infections. So their important point is for this one, protections and then cushioning, absorb the shock. So this is the most important point you need to write in your exam. Okay, I think that's all. And then for this one, artery in the mother, they will transport the oxygen blood. So it's a pulmonary artery, okay? Pulmonary artery will transport the deoxygenated blood from the heart side to the lung. Okay, and then for this one they show about umbilical artery. Okay, umbilical artery also same to the pulmonary artery. You look at this word artery, okay? You don't think this artery is mean what? It means it transport the oxygenated blood. Actually, this umbilical artery, right? They are actually transport the deoxygenated blood. This is for you to know, okay? Umbilical artery transport deoxygenated blood. Umbilical vein will transport the oxygenated blood. You can write down. Umbilical veins, okay, they will transport the oxygenated blood from the mother to the fetus, right? So the umbilical artery they will transport the deoxygenated blood from the fetus to the mother. So here's a pathway, okay? Here's a pathway. So what kind of waste is that urea? Okay, urea will come outside. They will get they will get from the fetus to the placenta to the maternal blood. Okay, so other kind of question you can just read by yourself. The answer is already there. Okay, so you don't need to waste your time to do it already. But I want you to study, right? You read on the answer, then you study how you're able to write the sentence. Okay, and this way, I hope by this way you can help. I mean, it can help yourself on how to write a sentence when you are answering the paper 4. Okay. So I think that's all for today. We end the class now, and then please remember to do your chapter 16 paper 2, and then I will see you next week. Bye.